Hello, my name is Twisha Sharma, and I'm a junior at Valley Christian High School. I'm deeply interested in genetic research, especially regarding autoimmune diseases. I believe analyzing the genetic basis of diseases provides a promising avenue for medical advancement. I wrote a research paper analyzing the variants of the PAX8 gene and how they impact protein structure and function in reference to a disease known as Hashimoto's thyroiditis, under the guidance of Cambridge University Research Associate Dr. Vera Kotseva. Currently, it is under the process of publication, and it was accepted to be presented at the Butler Undergraduate Research Conference. Aside from this, I'm part of an activity where I do research and also provide mechanical engineering support for a team that designs experiments to be sent to space. Moreover, I'm the vice president of the Global Impact Council, an organization that teaches and supports teenagers to create positive impacts in their communities, not just in the US, but in over 7 different countries. I also founded the Scientific Soiree, a study website and podcast dedicated to breaking down complicated scientific theories into an easy to understand format for students. Alongside these, I privately tutor fellow classmates in science classes and am currently serving as the founder and co president of the pre-med club at my school. My area of study is biology, and I aim to become a surgeon in the future. Today, I want to present my research on the genetic involvement of the PAX8 gene in the development of Hashimoto's thyroiditis, which is a chronic autoimmune thyroid gland disease. Hashimoto's thyroiditis is the leading cause of hypothyroidism in the United States, impacting millions of individuals, predominantly women, and causing a wide range of symptoms including fatigue, weight gain, and endocrine imbalance. Despite its high prevalence, the precise genetic mechanisms underlying this disease remain largely unknown, which prevents the likelihood of accurate diagnosis and specific therapy. My study specifically focuses on PAX8, a transcription factor that plays a critical role in the development and regulation of the thyroid gland. Mutations of the PAX8 gene have been implicated in many thyroid diseases and may play a significant role in the initiation and progression of Hashimoto's thyroiditis. This project follows a computational path in identifying and classifying PAX8 variants according to publicly available datasets such as ClinVar and alpha-missense. Other than that, I also created molecular models to ascertain the structural impact of such mutations on the PAX8 protein by utilizing software such as AlphaFold, PyMol, and Inkscape. The aim of this research is to enhance our understanding of the disease-causing mechanisms of PAX8 mutations. By doing so, it also contributes to the general goal of developing more targeted diagnostic methods and personalized treatment strategies for individuals who have Hashimoto's thyroiditis. As Hashimoto's thyroiditis remains substantially underdiagnosed in many parts of the world, this research also addresses a major global health issue by ushering in the genetic foundation necessary for more effective and equitable delivery of healthcare. Overall, research done from a genetic standpoint helps tackle the root of the problem in the body and has the potential to improve global health as a whole. From the larger set of genes, I selected PAX8. I then isolated 33 exon variants of the PAX8 gene, i.e., mutations in the coding region of the gene. These were filtered using alpha-missense, an artificial intelligence tool that predicts whether a mutation is likely to be pathogenic or benign. By comparing the outcome of ClinVar and alpha-missense, comparing effectively clinical outcomes with predicted outcomes, I was able to compare how clinical outcomes and AI predictions agree or disagree on classification for each variant. To determine the likely structural impact of these mutations, I built molecular models from the structural data from Protein Data Bank and AlphaFold, the two largest protein modeling databases. I rendered the protein structures in PyMol and colored every mutation with color codes for pathogenicity using Inkscape for visualization purposes. The molecular model in this case is the known PAX8 structure from Protein Data Bank with the changes above. I discovered within this analysis that 62.5% of the pathogenic mutations had taken place within the PAI subdomain of the paired box domain of the PAX8 protein. This region is necessary for DNA binding and gene regulation, and, as such, mutations in this region presumably render the gene unable to induce thyroid-specific action. Fewer pathogenic variants had appeared within the RED subdomain, which also assists in DNA recognition. Importantly, ambiguous or conflicting interpretation variants were more frequently encountered outside of the established protein structure, namely in intrinsically disordered regions. This understanding allowed me to apply a secondary classification approach, where variants located in alpha helices, the building blocks of the PAX8 protein structure, were anticipated to be more likely to be pathogenic, and in those disordered regions were most likely benign. These findings solidify the connection between PAX8 structural integrity and the thyroid functionality and offer possible explanation as to why one patient will develop Hashimoto's and another will not, based on certain genetic factors. My research also places emphasis on distinctions between computational and clinical classifications and highlights the importance of synthesizing both AI predictions and clinical factors for enhanced understanding of genetic disease. In summary, this presentation has explored the role of PAX8 gene variants in the development of Hashimoto's thyroiditis with the view of understanding better how mutations in genes can lead to the failure of protein structure and degeneration of thyroid function. Applying a computational and structural methodology, I examined 33 exon variants of the PAX8 gene. I contrasted clinical classifications with the ClinVar database with AI predicted classifications from alpha-missense and constructed detailed molecular models for visualizing the location and probable effects of these mutations. Of significance was the fact that slightly over 60% of the pathogenic variants were mapped to the PAI subdomain of the PAX8 protein, and this is a region critical to DNA binding. Also surprising was the fact that variants predicted as uncertain or benign had a tendency of occurring in disordered or less structurally important areas. This suggests that the structural location of a mutation may be just as important as the mutation itself when determining pathogenicity. These findings highlight the significance of integrating AI, clinical data, and protein modeling together using the integrated bioinformatics tools to gain more insight into genetic diseases as well as to guide the development of more targeted diagnostics and therapies. The results, however, are limited in that experimental confirmation was not performed within the context of this paper. Also, AlphaFold models may not be entirely accurate, as they are predicted by an AI. Shown above is the per residue model confidence score of the downloaded protein. This also suggests that Hashimoto's thyroiditis is affected by the PAX8 protein's ability to bind to DNA. As we move toward a future of personalized medicine, studies like this raise an important question. How many other underdiagnosed conditions might be better understood and treated if we more deeply explored the genetic and structural basis of disease? Finally, I would like to thank Dr. Vera Kozheva for guiding and mentoring me to write this research article. I also give many thanks to the Cambridge Center for International Research for assisting me in the publication process. Thank you so much for listening to my presentation.